Hello my dear students. So now we will be discussing artificial vegetative reproduction of plants. In the first chapter I discussed what reproduction is. So it is the life process that is needed for the existence or continuity of life. Under that, organisms can reproduce by either asexual methods or sexual reproductive methods. Thereafter, we discuss the natural vegetative reproduction shown by plants. So, that is the asexual reproduction shown by plants, and we considered it as the natural reproduction shown by plants. So, now I am going to discuss the artificial vegetative propagation. When we discuss the vegetative reproduction of plants, I told you all, if it happens naturally, then it is going to be the natural vegetative reproduction. But here we will use one of the vegetative parts in order to reproduce the plant. Then of course we call it the artificial vegetative reproduction or artificial vegetative propagation. Production of plants vegetatively by man is known as artificial vegetative propagation. So, production of plants vegetatively by man is known as artificial vegetative propagation. So, propagation is again producing new plants, making them to grow. So, that is done artificially by man. So, that can be done in different ways. We can carry out artificial vegetative reproduction by rooting of stem cuttings, by the method called grafting. Then there is the method of layering and also tissue culture. So, there are 4 different methods. So, this can be done in several ways. One is rooting of stem cutting. Then there is the method of grafting, layering and tissue culture. We will discuss these methods one by one. The first one, rooting of stem cuttings. So, when we say rooting of stem cuttings, I am sure students you all would have heard of this before also. Now, mostly the plants that do not reproduce sexually, or the plants that do not normally produce fruits and all that. Plants like roses, shoe flower, what do? We have the mature plant, we cut a part of the stem and we plant it somewhere. So, there the roots form and that grows into a new plant. So, if you have a stem like this from the mature plant, you have the stem, we make sure there are no young or immature leaves or fruits or flowers. So, then we cut a part of the stem and we replant it somewhere. So, this is what is called as a stem cutting. And so, when we plant it, there is root formation. So, that is why we call this method as rooting of stem cutting. To make the roots to form more efficiently, we can also use the growth substances. So, now we will see how this is done or what is rooting of stem cuttings is. Here you can see when we say rooting of stem cutting, new plants can be obtained by planting stem cuttings of a mother plant. So, as I showed you all, when the mother plant is there, from the mother plant we select a twig that is part of the stem, we cut that and we make sure there are no young leaves or flowers or fruits. So, then we cut that stem and we replant it somewhere where it forms roots and grows into a new plant. It is more appropriate to select twigs. So, this is what I told you all. Select twigs from a healthy plant. One thing, it has to be healthy, that do not bear tender leaves, flowers or fruits at that time. So, another point that you need to remember. So, one thing, we have to select a healthy plant. Otherwise, there is no point of replanting it. Why? If it is an unhealthy plant or an infected plant, then it may not survive and grow into a new plant. So, you have to select a healthy plant, and also we have to select the twig that does not bear tender leaves, young leaves, or flowers or fruits at that particular time. So, it's okay to have the mature leaves in the plant. So, we cut the stem cutting and replant it. These are some examples. You can see this is how they plant the rose plant. So, same stem cutting. This is of course after it has been planted, they are like actually making the soil bed and all that. So, stem cutting of rose. Then there is shoe flower, exora. Then we have bougainvillea and the croton plant. So, these are all done by rooting of stem cuttings, or examples of plants that can be propagated by rooting of stem cuttings. Rose, shoe flower, exora, bougainvillea and croton. I am sure you all can understand this method easily. It is a very simple method, students. You might have even done it at home. Cut off a part of the stem, that is a twig. You have to select the twig from a healthy plant, and the twig should not bear tender leaves, fruits or flowers at that time. Then you plant it at a new place where there is fertile soil, and you provide all the required conditions, and then there will be roots produced, and that will grow into a new plant. So, that is the first method, rooting of stem cuttings. With that, I will move on to the next slide. Here we have an assignment. Find out and record various agrochemicals in the market that are used to promote rooting of twigs. You are familiar with these, students, rooting of twigs. You all have studied about different types of plant growth substances in your previous grade. Can you think of some? Auxins, gibberellins, cytokinins. So, out of these, auxins are available naturally as well as they are available as artificial growth substances. Out of that, if you can remember, under auxins, IAA, indole acetic acid, acetic acid, and IBA, indole butyric acid. So, these are types of auxins that are used to induce rooting in stem cuttings. So, you all can remember that. This is the name of the auxin, the artificial auxin or artificial growth substance, IAA and IBA, but they can be sold by different names in the market. So, that is what you need to find out. So, when they are used as agrochemicals, you all know there are different brand names. So, they want you to find that out. Find out and record various agrochemicals in the market that are used to promote rooting of twigs. Then prepare a list of plants that cannot be propagated by stem cuttings. That also you all can think, students. Some of the plants that we discussed under vegetative reproduction, that is the natural vegetative reproduction method, and some that we will be discussing after this under sexual reproduction of plants. So, those plants cannot be reproduced by stem cuttings. Some of them can be reproduced in many different ways. They do produce flowers, they do produce fruits, and also they can be reproduced by stem cuttings. But there are plants that cannot be reproduced. So, you have to make a list of those plants. Then we have the next one. Investigate the features of a twig that should be selected for quick rooting. So, as we discussed before, one thing, the twig has to be from a healthy plant so that the new offspring, the new plant will also be healthy. Then it should not bear tender leaves or fruits or flowers, because then of course it won't grow into a new plant in a healthy manner, and also already there are fruits and flowers. You all know flowers are the reproductive structures of plants and the fruits are formed during reproduction. So, if you cut that and replant it, then the sexual reproduction of plant will be disturbed. So that is the reason why we should not select twigs that bear tender leaves, fruits and flowers. Tender leaves, why? Now only they are getting adapted and they are starting to do photosynthesis. So that is also not very suitable. So what should the twig be? It can contain mature leaves, a mature twig, healthy one. So like that, you can think of different features that need to be there in the twig that should be selected for quick rooting. If it is too thick, too rough, then also it might not root quickly. So you have to think of features like that. I am sure you all can do that. So you can carry out this assignment, find out and record various agrochemicals in the market that are used to promote rooting of twigs. Prepare a list of plants that cannot be propagated by stem cuttings, and also investigate the features of a twig that should be selected for quick rooting. So that is what you need to do here. With that, students, I will move on to the next slide. This is the next process. So there are 4 different processes under artificial propagation, artificial vegetative propagation of plants. First one was the rooting of stem cuttings. Then we have the second method, that is layering. Initiate rooting while it is still attached to the mother plant. So here what we do is initiate rooting while it is attached to the mother plant. In the previous one, rooting of stem cuttings, you cut the twig, you replant it, then only the roots form. But here, of course, we initiate the rooting when the twig is attached to the mother plant. So the twig is getting all the nutrients, food, everything from the mother plant, but there are roots formed. So then if you cut that twig and replant it, it can easily adapt to the environment and grow into a new plant. So that is the difference between layering and rooting of stem cuttings. In the first process, that is rooting of stem cuttings, roots are formed after replanting the twig. But here, rooting is initiated when the twig is attached to the mother plant. Then we replant it. So, different order. Initiate rooting while it is still attached to the mother plant is known as layering. And layering can be done in 2 methods depending on the twig that we select. If the twig is closer to the soil level, ground level, then it is done in one way, that is known as ground layering. And if the twig is high up in the plant, somewhat far from the ground level or higher from the ground level, then we call it aerial layering. So, 2 methods of layering. One is ground layering and the other one is aerial layering. 2 methods. Now we will discuss these methods one by one. The first method, ground layering. So here first look at this picture, students. You can see. Now when we select a plant for ground layering, one thing, there should be twigs that are closer to the ground level, and also the twig should be somewhat flexible so that you can bend it like this. When you try to bend, if the twig breaks, then of course you can't do this method. So you have to select the plant where the stems can be bent and also they have stems closer to the ground level. So this is what we do. We select a stem again from a healthy plant. It has to be a healthy twig, and here also we make sure the twig does not have immature leaves or very tender leaves, flowers or fruits. We select a twig with mature leaves and all that. That has to be healthy as well. Then what we do is we remove a layer of the bark. Now if this is the stem, from here we remove a part of the bark from the bottom, because this is going to be done with the ground. So if you remove a bark only from the bottom side, that should be enough. So here of course the vascular tissue, the xylem, phloem, they are actually exposed to the environment now. Normally the bark is the outer covering. So when you remove the bark, inside you have the vascular tissue. So that is now exposed. That actually helps in the formation of roots. So there is food being transported, and when that is in contact with the soil and all that, with the food and water, the root initiation is more faster. So then what happens is, if we look at the method, first a small cut is made on the underside of the selected twig. So as I told you all, you select the twig, then you make a cut at the underside. So a small cut is made on the underside of the selected twig. Then what we do is, we dig a very shallow hole in the soil surface, or just spread out the soil. Then you keep that part of the twig with this bark removed, and over that again you put soil like this and you cover it up. And also you can see this U-like metal structure, a metal piece like something similar to a clip-like structure. If we press that down, now if you just bend the twig and if you cover it with soil, the soil is not very strong to hold the twig in that position. So the twig can come off. To prevent that, what we do is, now if the twig is this way, we put this U-shaped metal like a hook. We push it down with the twig. It has to be around the twig, and that is also pushed into the soil. So then that will keep the twig in place. Then we need to water it, not a lot of water, just a small amount of water, so that the soil does not dry off too much. Then of course you leave it like that for a few weeks. After a few weeks, if you pull out the twig, you can see the formation of roots. There will be roots formed. Then of course you can make a cut at this point, and you can replant it somewhere else. You can allow it to put a cut, then this twig will go back to this plant, this will grow into a new plant, or you can take it, uproot it and replant it somewhere else. So that is how ground layering is done. If I go through the steps again, first a small cut is made on the underside of the selected twig. Then the twig is bent and buried under the soil. So that is what I told you. You take out a little bit of soil and bury it under the soil. After few weeks, the twig will develop roots. Then the twig is separated from the mother plant and is planted. So you can separate the twig and replant it. Examples: jasmine and lemon. You all can remember these plants, students. Jasmine with white color flowers and lemon. Not all lemon and lime can be used for ground layering. They should be the species where you have this twig that can be bent down like that. If the twig is too strong, then it's difficult to do the ground layering. Maybe we can go for aerial layering. So I am sure you all can understand this method, students. So here we have discussed the method of ground layering. So this is what you do. Select the proper twig, then you put a cut at the underside of the twig and you remove the bark, or you can just put a small cut and then you bury it under the soil. You use that pin-like structure to hold the twig in place. Allow it for a few weeks until root is initiated. Then you can take out the twig and you cut off the twig from the mother plant and you can replant it. And when replanting also, it is better if we take the twig with a small amount of soil. You all know that. Without just pulling out the twig with the roots, it can damage the roots. So with soil, if you take it out, then you can replant it. That will grow into a new plant. So as you can see, these are all vegetative reproduction methods or vegetative propagation method, sexual reproduction. So here you will get a new plant identical to the mother plant, more or less similar to the mother plant. That is what we discussed under asexual reproduction. So with that, students, I will move on to the next slide where I will discuss the aerial layering method. So aerial layering. As I told you all, if the twig is closer to the ground level, we can go for the ground layering. But here you can see the twig is somewhat above the ground. So, it's at a higher level. Then you can't bend the twig and do the layering process. Then we go for the aerial layering. So, this is what we do. We have the mother plant. Again, we select a twig. A healthy plant is selected. From that you select a twig again without tender leaves, fruits or flowers, and then you remove a bark like a ring. Again, like earlier, you remove the bark. So, like a ring. So, here we remove the bark. So, this is how the twig is going to be. At this point where the bark is removed, now there is phloem here and phloem here. This part has been removed. We have to make sure we don't damage the cambium and all that. We will discuss that in detail in your next grade. Now, cambium is a tissue that is present in dicot plants, and usually they are responsible for growth and development, especially the secondary growth. You are familiar with that. Dicots show secondary growth, whereas monocots, they don't show secondary growth. So, when we are cutting the bark, we make sure that we don't damage the cambium or the xylem. But the phloem can be removed. Then what will happen is the food that is coming down from photosynthesis of the leaves will accumulate here. So, at this part there will be enough food, there will be enough nutrition coming from this side, from the stem. So, that part of the twig has all the required components. Then what we do is, we put some compost soil mixture over it, and around that we can place the coconut husk, or we can use a polythene to cover it up, and you put 2 knots over here. You tie it like tying a toffee. So, then we allow it to be like that for some time. There you will see the initiation of roots. So, once that happens, we cut this off and we replant it. So, that is again shown there in the diagram. This is how we put the cut. We remove the bark. Then here you can see the compost soil mixture, and also you can see it being covered and tied. And sometimes what they do is, if it is a very tall tree, if it's difficult for us to water this part of the twig, what they do is they attach a small container like a can or something here. Into that they take water. From there they put a twine and connect it to this part. So, through the twine there will be water oozing, and that will be given to this part of the twig. So, it will be kept wet, there is enough water, everything is provided. So, then of course there will be root initiation. So, then this part can be replanted. That is the process of aerial layering. So, if we try to understand that aerial layering, this method is used for the twigs which are high above the ground. A ring of bark. So, as I showed you all, a ring of bark of the twig is removed. So, a ring of bark is removed. A mixture of compost and coir dust is placed round that place and tied with a strip of polythene. So, we use a strip of polythene to tie it. And after few weeks, the twig will develop roots. Then the twig is separated from mother plant and is planted. So, that is the process. In pomegranate, lemon, these types of plants, the aerial layering is more suitable. Why? Pomegranate, you can remember students, the stem is somewhat strong. So, it's difficult to bend the stem and do the ground layering. Then of course, we can go for this method if it is high above the ground. Same thing for lemon. Lemon also I mentioned to you all, some of the species of lemon you can do ground layering. Some you might need to do aerial layering. Sometimes in the same plant, if there is a twig closer to the ground level, you can go for ground layering, and for a twig that is high above the ground, we can go for the aerial layering. So, both methods can be sometimes used for the same type of plant. Is that clear to you all? So with that students, I will move on to the next slide where I discuss some advantages of layering. So following are some advantages of layering. Now what are the advantages of layering? If a plant cannot produce seeds, then it's easy to carry out this method. So plants that do not produce seeds can be propagated successfully by this method. That is one advantage. Plants that do not produce seeds can be propagated successfully by this method. Then the next one, we can get many number of plants within a short period of time. So that is also another advantage. Can get a lot of new plants from one mother plant. So here we can say several number of plants can be produced. Several number of plants can be produced. So those are the advantages of layering. We saw what layering is, and there were two types of layering, ground layering and aerial layering, based on what type of twig we choose, and both these advantages are common for both types of layering. With that students, I will move on to the next slide. Activity: identify a plant in your home garden which is suitable for ground layering. Follow the process of ground layering properly. After about 2 weeks, uproot twig and observe how roots are developed. So you all know the method of ground layering. From your home garden, you have to select a suitable plant and carry out the layering process. So that is again an activity that you all have to do, students. With that, I will move on to the next slide. This is the next process of artificial vegetative propagation, that is grafting. So here, connecting a twig or a bud of a plant to a plant of same or closely related species is known as grafting. So here of course from the introduction you all can understand, students, there are two plants involved. Although we have discussed before that for asexual reproduction or vegetative reproduction of plants, we only need one mother plant. But here of course there are two plants involved. So connecting a twig or a bud of a plant to a plant of same or closely related species is known as grafting. So for example, if you take a rose plant, say a red colored rose with a white colored rose. So both of these plants can be grafted together. So there we can either use a twig or a bud, and there it is a plant from one plant we can do, or plant of same or closely related species. So then, as I told you, there are two plants involved. Two parts of the plants grafted are mentioned below. One part is known as the stalk plant. The rooted part of the plant is known as stalk. So the stalk is rooted to the soil. That will contain the root system. So there it has to have a very good root system, a healthy root system. It should be able to withstand the drastic environmental conditions or changes in the environmental conditions, and also it should be resistant to pests and diseases. So there we choose a plant as a stock that should have all these characteristics. So the rooted part of the plant is known as stalk, and following are the characteristics that should be shown by a stalk. So here this is the plant that is going to be rooted to the soil. So therefore it should have a very strong and healthy root system. That is one characteristic. So should have a strong, healthy root system. And also it should show uniform growth, or the root system, the stalk should have a uniform growth. So that is the second characteristic, having uniform growth. And then if this is going to be the plant that is going to be rooted to the soil and it's going to grow to the environment, it should be able to withstand the changes in the environment as well as diseases. So that is another requirement of this stalk plant. Withstanding environment changes, and diseases. So two plants are involved, or two parts are involved. One is the stalk, that is the part that is rooted to the soil. So because of that, it should have a strong, healthy root system. It should have uniform growth, and also it should be able to withstand environmental changes and diseases. Then the second part is known as the scion. So this is the twig or the bud. Again, it is a twig or the bud taken from another plant and grafted to a stalk is known as scion. So as we discussed before, it has to be a twig or bud of a plant that is grafted to another plant of a same or closely related species. So here we have this scion, that is the twig or the bud taken from another plant and grafted to a stalk is known as scion. Following are the characteristics that should be shown by a scion. So like how we expect the stalk to be. The first characteristic is that it should be a variety with good characteristics, desired characteristics. Then only the purpose of grafting will be satisfied. When I explain the process, then of course you will understand better, students. The scion has to have good characteristics. So here we can say it should have good characteristics. That is one. So if it has, let's say if there is a rose plant that is used as the stalk, it has a very good strong root system and all that, but it produces flowers where the petals are not enough, or the flower is not very big enough, or maybe it has a certain color and we want to graft another color with the same plant. Then the second color, that is going to be the scion, that has to have the good characteristics. Say if you want a red color, very large flower, those are the good characteristics. That should be there in the scion plant. That is the first requirement. Then of course, the scion has to be free from pests and diseases, because you are going to graft that plant to the stalk plant. So if this is already affected by a pest or a disease, obviously that will be given to the stalk as well, then the stalk also will be affected. So it has to be free from pest and diseases. So it should be free from pest and diseases. So those are the characteristics. So we are discussing the grafting technique, students. So here connecting a twig or a bud of a plant to a plant of same or closely related species is known as grafting. So in that two parts of the plants grafted are mentioned below. One is the stalk, the other one is the scion. Stalk, the rooted part of the plant is known as stalk, and following are the characteristics that should be shown by a stalk. It should have a strong healthy root system, having uniform growth and also withstanding environmental changes and diseases. Then if we look at the scion, twig or the bud taken from another plant and grafted to a stalk is known as scion. And following are the characteristics that should be shown by a scion. It should have good characteristics and it should be free from pests and diseases. So this is what we will be doing. Now you have a stalk plant. It has a very strong root system. Then you have the stem and maybe you have the twig. So this to this stem, if we put a cut there, this is going to be the stalk, and to that you are going to bring in neither a bud or twig, and that bud or twig is going to be attached there. So thereafter we have to wrap it with a strip of polythene and we have to allow it for some time, few days. So then after that, when the bud or the twig starts growing along with the stalk plant, then we remove the polythene and allow it to grow together. So here we have the stalk with the good root system and we brought in the scion. And based on whether the scion is a bud or a twig, we classify it into two types. One is known as the twig grafting, the other one is known as the bud grafting. So that is how we carry out the grafting method. So two types, twig grafting and bud grafting. Now let us discuss those two methods. In the process of grafting, stalk and scion are fused together by their cambium. This is very important, students. The stalk and scion, they have to be fused together by the cambium. Now, if I go back to the previous one, this is how I showed you all. This is where a bud is usually attached or a twig is attached. But in some processes, in a twig grafting process, you can have the stalk like this and we make a cut on the stalk. We bring the twig, that is the scion. This is going to be the stalk and this is going to be the scion. And when the scion is brought into the stalk, they have to fit with each other. Normally we graft two plants that are of similar age, so that they have similar diameter for the stem, then they will fit with each other. And you all know, if you take the cross section of a dicot plant, this has to be a dicot plant. In dicot plants, you all can remember, students, they show secondary growth. I have mentioned this before also. Secondary growth is possible because of the cambium tissue. You will learn about this in the next grade in more detail. There you can see in the stem, if you take the cross section, normally you can see the vascular bundles like that, the xylem and the phloem tissues. In that, in the middle, there is the cambium tissue. That is the tissue that is responsible for secondary growth. So this is the cambium tissue. The important thing is, when you have the stalk and the scion, they will both have the cross section like that. The cambium of the stalk and the cambium of the scion have to fit together. Then only they will start growing together as one plant. So this technique is possible only for dicot plants, because only in dicot plants we have the cambium. You all have to remember that, students. So with that, I'll again move to the next slide. So in the process of grafting, stalk and scion are fused together by their cambium. Very important. Therefore, grafting can be done only on the dicotyledonous plants. That's again very important, which have cambium tissues. Grafting can be done in two ways as follows, as I told you all, based on what we use. One is known as bud grafting. The second method is known as twig grafting. So, this is actually based on the scion. If scion is a bud, we call it bud grafting. If scion is a twig, we call it twig grafting. Under those methods also, depending on how the cut is made, how the grafting is carried out, there are different techniques. But we will just try to understand what bud grafting and twig grafting methods are. Bud grafting. Selecting a plant bud as the scion and grafting it to a stalk is known as bud grafting. So, you select a bud as the scion. Why bud? Normally, the buds are the parts of the plant that actually grow. Apical bud increases in size. Then axillary buds, you know, they form the branches. So, that is why we select a bud. So, selecting a plant bud as the scion and grafting it to a stalk is known as bud graft. And this is how that occurs, or it is done as follows. This is the method. Cutting a live bud which is located above the leaf scar using a grafting knife. So, few points you need to remember. The bud has to be live. It has to be from a growing or living plant, a live bud, and which is located above the leaf scar. What is a leaf scar? When the mature leaf falls off from the plant, from the branch or the twig, it leaves a scar. You can see the point where the petiole of the leaf or the stalk of the leaf was attached to the twig. That is what we call as the leaf scar. And we need to cut this using a grafting knife. Normally, these grafting knives are very sharp knives with curved edges, used specially for grafting technique, so that they don't damage the tissue. You can easily cut the bud off, and also you can attach it to the stalk plant. So, then making a cut on the stalk and insert the bud into the cut. So, we put a cut on the stalk and we insert the bud into it, and thereafter wrapping the place from bottom to top using polythene strips. So, it has to be wrapped from bottom to top using polythene strips, and we leave that for few days. After few days, when the bud is about to emerge, remove the wrap and re-wrap, keeping the bud open. I will explain all that. We have to remove the bud and re-wrap, keeping the bud open. The cutting the stem of the stalk about 15 cm above from the bud after about 3 weeks. So, that is the process. Here students, you all can see the process. Now, this is the scion. Here we have the stalk. So, here you can see a bud has been removed. So, near the leaf scar, where the leaf scar is, you use a grafting knife, and using that we cut off the bud, and at the same time in the stalk also there is a cut. Here of course you can see the cut is like a T-shaped cut. So, normally we will call this process as a T-buddy, because here the cut is like a T-shape. Then what we do is we insert this bud into this T-shaped cut. Here you can see the bud, that is the scion, has been inserted into the stalk. Then it's wrapped around like this. You can see here. Initially we cover everything completely. Now around this also the polythene is wrapped. We allow it like that for few days. Why do we need to wrap it? When you wrap it only, if you just insert it and let it be like that, the bud can fall off. But when you insert it into the cut and when you wrap it with a polythene, it will be intact, and the bud also will attach with the tissue of the stalk. Bud is the scion and the stalk is that plant. So they will attach together and they will start growing together. And thereafter, from this bud, there will be a twig that will start to grow. So at that point, when the twig starts to emerge only, we remove the wrapping, that is the polythene strip, and re-wrap it like this. So from here, always wrapping from, if that is the stalk, we start wrapping from bottom to top, bottom to top. That is how you are supposed to wrap it. Then after we remove the first wrapping, that is when the twig starts to grow. Again we re-wrap, but exposing this part to the environment. Now because it's already fused to the stalk and scion have become one and the scion has started to grow. So therefore we expose that to the environment and re-wrap it. So after this is done, what we do is we need to cut off the stalk. Now if this is the stalk part about 15 centimeter, if this is about 15 centimeters, we cut off the stalk above that. So only from here to here it is the stalk part, then from here it is the scion that is going to grow as the new plant. So we have the root system, that is the stalk, and the scion growing as the twig. So here we have fused or we have joined two plants together, the stalk and the scion. Then there are several types of bud grafting according to the shape of the cut on the stalk. So here I told you all this is a T-shaped cut. So there is T budding. If the cut is going to be an H-shaped cut, we call it H budding. And if it is V-shape, we call it V budding. So that depends on how we make the cut on the stalk plant. Is that clear to you all, students? So that is one method of grafting. This is bud grafting. So we have seen that bud grafting method. Stalk is the plant with the root or plant that is rooted to the soil, and here the scion is going to be a bud. And very important, it has to be a live bud which is located above the leaf scar. Using a grafting knife we have to cut it. So as I told you all, a grafting knife usually a sharp blade knife with a curve at the tip. So that is used for specifically for budding purposes. Is that clear to you all? So now that we have discussed the bud grafting technique, I will move on to the next slide where I discuss the twig grafting technique. Twig grafting. In this method, a twig of a plant is taken as the scion. So here twig is the scion. Twig is taken as the scion. Procedure of twig grafting is given below. So here again the procedure is shown to you. Selecting a twig of a fruit bearing plant. So here we select a twig of a fruit bearing plant. It is more suitable to select a twig which has no tender leaves, flowers or fruits. We have been discussing this before also. You can remember, students, even when rooting of stem cuttings or even layering, we normally choose a twig that does not have tender leaves, flowers or fruits. I explained that to you all before also. Cutting the twig without damaging the cut. So when you cut the twig, the surface, the cut surface should not be damaged. The tissues in the cut surface should not be damaged. Fixing the twig to the stalk so that the cambium are connected. Very, very important. The cambium should be connected. So I told you all before also, when we select the stalk and the scion, we have to make sure the diameters are somewhat similar. So if you select plants which are of equal age or maturity, then most probably their stems will be of the same size, the twig and the stalk. So then the cambium will get connected properly. Wrapping the place from again bottom to top using polythene strips, always from bottom to top. Removing the wrap when the twig is observed to be growing. So here the picture is shown to you. Here also there are different methods. Now here you can see this is the stalk and that is the scion, and that's a twig that is coming in. So you need to make a cut there, and when now say if we make a now in the stalk, if we make a cut something like this, the scion has to be also something like this. So the scion will fix into the stalk easily. So here again, based on the shape of the cut, we have different techniques. So when they are fixed together, now here you can see like this, like a wedge we have made the cut. So here the scion is fixed into the inserted into the stalk, and then you have to wrap the polythene from top to bottom so that the scion is kept in place with the stalk. So thereafter, once it's wrapped completely like that, it is allowed to grow, and once you see after few days, when you see or when you observe that the stalk and scion, the cambiums have fused together and they have started growing together, then of course this polythene strip can be removed. So then they will be grafted together and the new scion will grow. So we will have the root system of the stalk and the rest of it will be the scion. Here also, according to the shape of the cuts of the edges of stalk and scion, several types of twig grafting can be done. So as example, we can consider arch grafting, and also peg grafting. Arch grafting, from the name, students, you all can remember. You might have seen this in certain places. Two plants growing on either side of the road, they are brought together and connected at the top like an arch, and they grow together. So there one will be the stalk, the other will become the scion. So there we can do the arch grafting. Then peg grafting again, there are many grafts that are connected together. That is also another type, arch grafting and peg grafting. So this is what happens during grafting. So we use the twig as the scion, and also we make sure it is a fruit bearing plant, and when we select the twig, it should not have tender leaves, flowers or fruits. So after that we make the cut so that we don't damage the tissues, because we need the cambium intact, then only the stalk and the scion will grow together. So you insert the scion into the stalk, you wrap it with polythene strips and allow it to grow. So that is the simple procedure of twig grafting. These are the things that you need to remember. And also we saw that there are two methods of twig grafting based on how the cuts are made. So there can be arch grafting or peg grafting. I am sure you all can understand that technique now. With that, students, I am going to move on to the next slide. So here we have an activity. Try to perform a twig grafting or bud grafting with the assistance of your teacher. If a grafting knife is not available for this, use any other sharp knife. So as I told you all, grafting knife, there is a handle like that, and at the end there is a slide bent like that. So that is used easily to cut the twig or bud. So we can either perform a twig grafting or a bud grafting with the help of the teacher. So actually I am not there to help you all. You all can try this with the help of your teacher and try that and see. With that, students, I will move on to the next slide. So following are some advantages of grafting and budding. So grafting and budding, the twig grafting and bud grafting techniques. If we consider the advantages, what are the advantages? We can successfully propagate plants that do not produce seeds. That is one advantage. So here I can say plants that do not produce seeds can be propagated successfully. That is one advantage. Right. Then the second advantage. So we have the stalk plant, that is the strong root system. And to that we are going to attach the cyan. So we will get a plant with the characteristics of cyan. That is another advantage. So production of offspring with the characteristics of cyan. That is another advantage. Another advantage is that you can obtain plants that are resistant to pests and diseases. So there it is an advantage. So we can produce plants that are resistant to pests and diseases with a strong root system. So the cyan will be resistant to pest and crop diseases, and also there is a strong root system, that is the stalk plant, and both of them are combined together. So you can produce a plant that are resistant to pest and diseases with a strong root system. So those are the advantages. Plants that do not produce seeds can be propagated successfully. Then production of offspring with the characteristics of cyan, and also produce plants that are resistant to pests and diseases with a strong root system. So those are the advantages. At the same time, there are disadvantages. What are the disadvantages, students? One disadvantage is that these plants have a short lifespan. Why? We choose a stock of one plant, we choose the cyan from another plant, we combine them together. So normally they have a very short lifespan. So that is one disadvantage. Having a short lifespan. That is one disadvantage of budding and grafting. And also this is not very successful with all types of plants. So that is another disadvantage. Not successful with every plant. Another disadvantage is that it reduces the wood value of plants. It reduces the wood value of plants. So those are the disadvantages. Having a short lifespan, not successful with every plant, and reduces the wood value of plants. So those are the disadvantages of grafting and budding. So we have discussed the different techniques, students. Under artificial vegetative reproduction, we already discussed rooting of stem cuttings. Then we discussed the process of layering, and now I discuss the process of grafting. Under that, the bud grafting, that is what we call as budding, and the twig grafting technique, and we have discussed the advantages and disadvantages of budding and grafting as well. So, the next process or the next type of artificial vegetative propagation is tissue culture. So, in the next slide, I will discuss what tissue culture is. So, now we will discuss tissue culture. New offspring which are identical to the parent plant can be produced by cultivating any vegetative tissue of a plant in a culture medium under controlled conditions. Offspring thus obtained are called a clone. So, here you might have heard about cloning. From a cell of the organism or even a small tissue, a tiny part of the tissue of an organism, we can produce a new organism, and that will be identical to the mother plant. So, here we are considering the tissue culture or cloning of plants. Right. So, if we look at the explanation, new offspring, that you all know, we are trying to produce the new generation which are going to be identical to the parental plant. Exactly the same, because you are going to use the tissue from the parent plant. Can be produced by cultivating any vegetative tissue. It can be a leaf, it can be part of the root, it can be part of the stem, any part of the plant. Any vegetative tissue of a plant in a culture medium. Now, we have discussed this culture medium before also, students. You all can remember when we were discussing algae. One of the uses of algae is to produce or extract agar. And you can remember this agar is used to make the culture medium, actually to solidify the culture medium. So, in that culture medium, we can grow bacteria as well as we can carry out tissue culture. So, it's the same basic thing. When we say culture medium, you use that medium to grow something. Whether it's a microorganism or tissue culture, it's basically the same culture medium. But the nutrients or the substances added to that culture medium may vary depending on the purpose. So, here also we use culture medium, and there should be controlled conditions. Now, you all know for a plant to grow, plant to reproduce, it needs certain environment conditions. Now, these conditions have to be controlled here. Then only you will get the required offspring. Offspring thus obtained are called a clone. Why we call it a clone? Because it's going to be identical, exactly the same as the parental plant. So, then, if we look at tissue culture further, genetically identical clones can be obtained by tissue culture. So, this is also important. Genetically identical, because we are going to use the tissue from the parental plant. The same plant, identical plant will be produced, or identical offspring will be produced, and they will have the same genes. You all know about chromosomes, genes, the information that is stored within DNA molecules. It's going to be identical to the mother plant or the parent plant. So, in tissue culture, generally tissues are taken from the following parts. Now, you all can remember, students, in a plant, the plant grows. Normally, the shoot grows. So, it is the shoot apex that is responsible for the growth, increase in height of the plant. The same way, the root tip, that also grows. So, it can be shoot apex or shoot tip, root tip, and also from the lateral buds only in a plant, there are branches, flowers or fruits. They are all produced from the lateral buds. So, the shoot apex, lateral bud, and the root tip, those are the parts that are mainly responsible for growth. So, we try to select tissues from those parts so that they can grow easily and they develop into a new plant or a clone. So, generally tissues are taken from the following parts. So, here we can say the shoot apex, or we can say apical bud, apical bud. Then we can use the root tip, or even you can call it root apex, and also the lateral buds. So shoot apex or apical bud, then we can use the root tip or the root apex as well as the lateral bud. Those are the parts of the plant that are involved in growth mostly. So then the tissues can easily divide and form the new clone. So with that introduction, students, I will move on to the next slide. Substances included in the culture medium. So when we say culture medium, like a plant growing in the soil. So you all know for a plant to grow, it needs the soil. So that is the medium. And in soil, there is air, then there's water, there are nutrients, and it can inhale or intake oxygen as well as carbon dioxide from the atmosphere. So for the growth of the plant, it needs certain substances. The same things have to be provided through the culture medium. So we need to have a culture medium that is somewhat solid. So there, as I told you all before, we use agar to solidify the medium. But in addition to that, we need to include certain substances. One is sucrose. You all know what sucrose is. It is a disaccharide. So disaccharide made up of glucose and fructose. So from that, the growing tissue will get its energy. From carbohydrates, normally they act as energy sources. So here we add sucrose. Then we add nutrients. All the nutrients that are needed for the growth of the plant. So you all are familiar with that also. We have discussed minerals and nutrients. Then we need to add plant growth substances so that they will induce the growth of the plant. Plant growth substances. You all know they are also called as plant hormones. So we need to add sucrose, we need to add nutrients, we need to add plant hormones. I will write those down. So we need sucrose. So when I say nutrients, you all know two main nutrients that are needed. Apart from carbohydrate, proteins, lipids, the biomolecules, what are the nutrients? The minerals and vitamins. So here we will write them separately. Minerals and vitamins, as well as plant growth substances. Plant growth substances. So substances included in the culture medium are sucrose, minerals, vitamins and plant growth substances. And also there is agar. Agar is used to solidify the medium. So agar is a substance used to solidify the medium. Then we need to provide sterilized conditions, and controlling of temperature and light should be practiced for the success of tissue culture. Why sterilized conditions? Now I mentioned before also about the culture medium, using the culture medium for the growth of bacteria. So if you have all the nutrients, sucrose, minerals, vitamins and the culture medium, if the conditions are not sterilized, there can be microorganisms in the atmosphere, they can easily grow in this culture medium. So then you may not be able to get a pure clone, or the clone might not develop at all, and also the microorganisms will start growing in the medium. So to prevent that, we need to provide a sterilized condition. So sterilized condition in order to prevent growth of, the growth of microorganisms. Like bacteria. So sterilized conditions are needed. Then controlling of temperature and light. you all know when the seeds are growing normally, there needs to be a particular temperature, optimum temperature, so that the growth will take place efficiently. So, in order to do that, we need to control the temperature. We need to provide optimum temperature. So, this is one, we need to provide optimum temperature. So, this again will depend on the nature of the plant that you are trying to reproduce, and then even light. It shouldn't be too dark, it shouldn't be too much of light. So, we need to control the amount of light as well. So, sterilized conditions and controlling of temperature and light should be practiced for the success of tissue culture. So, you all have to remember, students, we need a culture medium. Into that, we need to add certain substances, sucrose, vitamins, minerals and plant growth substances. There should be agar in order to solidify the medium, and the conditions has to be very, very sterile. No microorganisms at all. And also, we need to have control conditions, especially the temperature and light. So, those are the requirements. So, this has to be carried out by well-trained personnel under very sterile environment, a proper laboratory. So, now we will discuss how this process is done. Following are the principal steps followed in tissue culture. So, following are the principal steps followed in tissue culture. Introducing the part of vegetative tissue obtained from mother plant into the culture medium. So, initially we need to introduce the vegetative part that is obtained from the mother plant into the culture medium. What are the vegetative parts? Shoot apex, root apex or lateral bud. Then what do we do? Allowing new roots and buds to develop from the tissue called callus. This is something you need to remember. So, the callus is the tissue that you obtain from the parent plant. That is what we are trying to develop into a new plant, which is grown from the vegetative tissue introduced. So, you take a vegetative part, you introduce it to the culture medium, then we call it the callus. That will develop into the clone. So, the terms are important. Separating the plantlets. What are plantlets? The new plants that are trying to develop. So, here it is not a seed. From a tissue, we are trying to generate a plant. So, that new structure is what we call as the plantlet, and placing them in test tubes or flasks to grow further. So, initially we have the culture medium, then we transfer it into a flask or a test tube, and thereafter gradually let the new plantlets to get adapted to the natural conditions to be cultivated in the field. So, then we transfer it into a soil containing tray, and thereafter it will be transferred to the natural environment. So, we will look at the steps again here. Now, here you can see this is a carrot. So, carrot means it's a root tuber. That is where the food is stored in roots, taproot. So, from that, so you can consider this as the root apex, so root part of the tissue. So, there we take a part of the tissue there, and here you can see this is the tissue that is going to be used. Tissue that is used. Here we have the culture medium. So, we have the culture medium there. On that we introduce the tissue and then it grows. This is where we call it as the callus. This will be known as the callus. So, those are the terms that you need to remember, students. Once the callus starts developing, we transfer it into a conical flask. Here it is a conical flask. Or you can transfer it into test tubes. Again, depending on the nature of the plant, how we need it. Here you can see from a very small amount of tissue, you can see so many new plants being produced. So, that is where you have the plantlets. Now, here you can see the plantlets. These are the plantlets. Plantlets. So here they have the parts that are needed to produce the root system as well as the shoot system. Then we transfer it into a natural environment. So this is similar to a natural environment because here you can see there is soil, but still somewhat like a similar to a culture medium, but thereafter we transfer it into trays. So this is almost like the normal environment. But even here, we keep it near the lab. Now, if you are carrying out the cloning process or the tissue culture process in a lab, this is part of the lab. Still, it has not gone on to the natural environment completely. So, here you can see once the plantlet starts developing, they are transferred here. This is almost like a plant. So, from the callus, we get the plantlets. These are the plantlets. Then they are transferred into the natural environment or natural conditions. So again, if we look at it, we have the normal tissue. We take the tissue, grow it in the culture medium, then it is known as a callus that is transferred to a conical flask or a test tube, and then that develops here. You can see this is enough to form the root system and the shoot system. Then it becomes the plantlets, almost the plants now. Then the plant is transferred to natural environment conditions. So up to this point, it is going to be inside the environment or a laboratory, we can say, tissue culture laboratory. So there all the conditions are controlled up to this point. But from here to here, there is a change. So that is where the plantlets adjust to the natural environment. And when they start growing here, the natural soil exposed to the atmosphere, sunlight, everything, they can be separated and replanted. That is how we grow the new offspring during tissue culture. Now, in these students, you all can see we took a very small amount. You all know the size of a petri dish, a very tiny amount of tissue. From that, you can produce a very large number of plants. And also that can be done in a very short period of time. So, in that manner, there is lot of advantage here. You can produce a large number of plants, new offspring within a very short period of time using a very small amount of tissue. But the thing is, all the plants, all the offspring will be exactly like the parental plant. So, because they will have the same genetic material, they will be exactly the same. That is why we call them clones. So, that is the term clones because they have the same exact DNA. Is that clear to you all? So, that is the simple explanation for tissue culture. So, you can see these are the steps involved, but when the process is carried out, the conditions have to be satisfied. It has to be carried out by trained personnel. With that, I will move on to the next slide. Following are some advantages of tissue culture. So, when we say advantages of tissue culture, production of offspring which are identical to mother plant. So, here identical to mother plant, that is one advantage. So, if you want a large number of plants which are identical, this is the best method. Propagating a large number of plants at the same time. So, I told you all a small tissue, a small part of the tissue, from that we produce a large number of plants. Then production of a large number of plants in a short period of time. That is another advantage. Large number of plants in a short period of time. Then propagation of a large number of healthy plants in a limited space. So, that is another advantage. So, when I said lab or before that, if you can remember looking at the steps, we have a small petri dish. From that, if they can produce about 100 plants. So, that petri dish is a very tiny space. From there, you are moving it to a conical flask. From the conical flask into a small jar. Then from there, it goes to a tray containing the natural condition, soil and all that. There you will have a very large number of plants. So, healthy plants in a limited space. Within the lab, you can produce a very large number of plants and can obtain a large number of plants using a hybrid tissue with a favorable gene. That is very important. Using a hybrid tissue with favorable genes. What are hybrid tissues? From similar species, you can select two plants with desirable characteristics. So, those desirable characteristics can be combined together in tissue culture. So, hybrid tissue. Both tissues are combined. And then from there we can get the favorable gene. All the desired characters can be obtained. So, that is an advantage in tissue culture. Is that clear to you? So, we can produce offspring identical to the mother plant. A very large number of plants can be produced, very large number in a short time, in a limited space, and they are all healthy offspring, and also we can get favorable genes using hybrid tissue. So, these are the advantages of tissue culture. But as I told you all students, this cannot be done by everyone. They have to be trained or skilled to do this process, as well as we need to have the proper sterile environment with control conditions. So, that is the requirement. Only thing is, here you will get plants that are identical to mother plant. So, that is in a way an advantage, but at the same time, we will have no variations. There will be no new characteristics produced. So, that might become a disadvantage as well. So, with that, I will move on to the next slide. Assignment. Prepare a list of places where tissue culture is practicing in Sri Lanka using various sources. So, here students, you all can look at the internet, you can read the paper, you can look at magazines or scientific articles. Then you can get information regarding places where tissue culture is practiced in Sri Lanka. Normally, the agro-biotechnical laboratories. So, we call them the agro-biotechnical laboratories. So, in agriculture and biotechnology. So, that's why agro-biotechnical laboratories usually carry out this cloning process or the tissue culture process. So, you all can find out what places carry out these processes. If possible, visit such a place and study the process of tissue culture. So, if you go there, you can have a look at the process and how it is being done. Name the plants which are mostly produced by tissue culture in Sri Lanka. So, if you visit a place like that, you will see what type of plants they produce. So, it again depends on the organization, the laboratory. Depending on the requirement, they will be carrying out tissue culture with different types of plants. So, all these have to be done by y'all. Y'all have to try and find this information. I am sure you can do that by yourself. So, with that students, I will move on to the next slide. Advantages of vegetative propagation. So, under vegetative propagation students, you all can remember we started off the lesson with reproduction, under reproduction, asexual reproduction and sexual reproduction. So, when we say asexual reproduction, when we consider plants, it's the vegetative propagation. And under vegetative propagation, we discuss the natural vegetative propagation as well as the artificial vegetative propagation. Under artificial vegetative propagation, you all can remember the methods: rooting of stem cuttings, grafting, then we discuss the layering and also tissue culture. So, 4 different methods. Under layering also we discuss aerial layering and ground layering. So, all these methods are considered as the artificial vegetative propagation. So, now here we are going to discuss the advantages of vegetative propagation. So, when we just say vegetative propagation, it includes both natural and artificial methods. So, here, first one, ability of propagating plants that do not produce seeds successfully. So, you all know students, there are plants. For example, rose, shoe flower, or even herbs and all those plants. These plants that do not produce seeds successfully, then they cannot have the sexual reproduction successfully, because you all know flowers and seeds are the important parts or the parts of the sexual reproduction. So, if plants do not produce seeds successfully, then they cannot carry out sexual reproduction successfully. So, they need vegetative propagation. It can be natural or it can be artificial. Then ability of having offspring that are identical to mother plant. So, identical to mother plant, that is another advantage. Like we saw in tissue culture, identical to mother plant. Even stem cuttings, you cut the stem from the mother plant, replant it. So, it's going to be identical. Again, when it comes to layering, they are also, the plants are going to be identical. But in grafting, we will be mixing characteristics. The stalk will have the characteristics of the root system will be of the stalk, and the scion will be the shoot system. So, there we will be mixing characteristics of two plants, root system and the shoot system. But again, in tissue culture, it's exactly the identical plant. The same goes for the natural vegetative reproduction also. Stem tubers, runners, all these are going to be same as the mother plant. Then, propagation of plants that bear fruits early. Now, there are some plants that bear fruits early. So, they carry out the sexual reproduction, but at a very early stage. So, thereafter, if you want to propagate them, then we can use one of these methods, the vegetative reproduction method. Then, ability of propagating selected plants which are resistant to diseases and pests. So, that is again another thing, which are resistant to diseases and pests. So, you can remember when we do grafting, we select the stalk and the scion in a method that they are resistant to change in environment conditions, they are resistant to diseases, they are resistant to pests. So, we select the suitable characteristics. That is possible during vegetative reproduction. Then, ability of generating plant varieties which withstand adverse environmental conditions. So, we can generate plant varieties that withstand adverse environmental conditions. So, we can produce, we discussed the hybrid tissue, desired characteristics. So, there we can develop new varieties, generating plant varieties, and also we can select the good characteristic, the good plants or the good varieties which have the desired characteristics, and we can propagate them. So, we can select plant varieties which withstand adverse environmental conditions. So, that is possible. Those are the advantages of vegetative propagation. But we have a disadvantage of vegetative propagation as well. One disadvantage. What is that? Here, we either need just one plant, except for grafting, we take two plants, otherwise we only use one parental plant. So, it's going to be the same characteristics. There is no meiosis. You can remember that we discussed that with the differences. No meiosis taking place. If there is no meiosis, there is going to be no mutation in genes. So, if there are no mutations, there are no variations. No variations means new characteristics are not going to be produced. Therefore, no new species can be produced. So, that is going to be the disadvantage of vegetative propagation. Disadvantages of vegetative propagation: new varieties are not evolved. Very important. Whether it's natural vegetative or artificial vegetative, in both propagation methods, new varieties are not evolved. So, with that, students, I am sure you all have a very good understanding about the asexual reproduction of plants. We discussed the natural sexual reproduction or the vegetative reproduction, or what we call as the natural vegetative propagation of plants, and then the artificial vegetative propagation of plants. So with that, I am going to end this chapter, and in the next chapter, I will discuss the sexual reproduction of plants.